Let's explore how linked genes affect inheritance patterns in A-level biology.
Recap from the previous lesson. Mendel's dihybrid crosses, F2 phenotype ratio is 9:3 to 3:1. The test cross ratio is 1:1 to 1 to 1 to 1 F2 phenotypes. He obtained 9 round and yellow peas and 1 wrinkled green pea. These exactly resembled their parents. He also had 3 round green and 3 wrinkled green. These two are different from their parents and are known as recombinants. These recombinants are formed as a result of the mixing up of genes caused by independent assortment and segregation with the test cross. He also obtained all the combinations in the ratio of 1 to 1 to 1 to 1 to 1. Today, we are going to discuss a situation where no new characteristics are formed, no mixing up of genes, no new combinations. There is no independent assortment and segregation. This is known as linkage. Genes are attached to the same chromosome-exclusive content for subscribers. Linkage deviates from Mendel's ratio. In Drosophila, the broad abdomen is dominant over the allele for the narrow abdomen. Then long wing is dominant to the allele for the vestigial wing. He crossed a broad abdomen with a long wing and with narrow abdomen with a vestigial wing. He obtained a different ratio. The F2 phenotype ratio is 3:1. He obtained 3 broad abdomens with long wings and 1 narrow abdomen with vestigial wings. These all resembled parents without new combinations. This also happened in the test cross with a 1:1 ratio. Mendel's ratios are not obtained. The genes on the same chromosomes form a linked group. You can also get more information from the self-study A-level biology textbook, competency-based. Let capital B represent the allele for the broad abdomen. Let small b represent the allele for the narrow abdomen. Let capital L represent the allele for the long wing. Let small l represent the allele for the vestigial wing. Parental phenotypes. Broad abdomen with long wing crossed with narrow abdomen with vestigial wing. Parental genotypes. We use a chromosome and attach the chromosomes. This is linkage. Genes or alleles are attached to the same chromosome. Crossed with double recessive characteristics. Meiosis leads to gamete formation. Gametes are formed. All the genes, alleles, go within the same gamete. This is called linkage. This also happens on the other side. Since all the gametes are the same, for simplicity, we can represent using one gamete, including the word all. Random fertilization to form the F1 genotypes. All will be F1 phenotypes, all broad abdomen with long wings. Since broad abdomen and long wings are dominant. Let a comment and a question. Intercrossing F1 genotypes to form the second filial generation. Parental phenotypes. Both have broad abdomens with long wings. Parental genotypes. Both are heterozygotes. Meiosis leads to the formation of gametes. Gametes are formed. Random fertilization to form the F2 genotypes. We cross and we obtain the following combinations. F2 phenotypes. The first 3 are all broad abdomens with long wings. The last combination narrow abdomen with the vestigial wing. This is the 3:1 ratio for the F2 phenotype. Linkage complete dominance. This deviates from Mendel's 9:3 to 3:1 ratio. Before we continue with the test cross, comment with the name of the school where you are watching the lesson. Leave a question for me. In complete linkage, a 3:1 ratio is obtained in the F2 generation because independent assortment and segregation of chromosomes do not take place. In a test cross, we use a heterozygote with double recessive. Parental phenotypes, broad abdomen with long wing, crossed with narrow abdomen, vestigial wing, parental genotypes. Heterozygous with double recessive. Meiosis leads to the formation of gametes. Having the number of chromosomes happens, random fertilization occurs to form the offspring genotypes. We do not call these filial generations because they are not obtained from pure breeding individuals' offspring phenotypes. The first two are broad abdomen with long wings. The second two are narrow abdomen with vestigial wings, making a 1-to-1 ratio. This is different from the test of Mendel's dihybrid. In practice, the F2-3-to-1 ratio and the 1-to-1 test cross ratio are never achieved. 4 phenotypes are invariably produced. This is because the total linkage is rare. Watch the next video to understand why. Thank you for your time and attention. If you like this video, please subscribe to the channel and explore our playlist for more advanced biology topics. Your feedback and questions are always welcome.